Good afternoon, or evening, or late night, or early morning, or whatever time it happens to be when you're listening to this. One of the advantages of distance learning is this ability to come to class, in quotations, when you're able, when it works for you. Before we jump in, let me pass along. A student just asked a great question, and the question was, how should I be studying for lecture exams? My suggestion is that you continue to study for these exams in the same way you would if you were going to be taking a regular on-campus exam. And if you know the material, you'll be fine. So just keep on doing what you would normally be doing, what you have normally be doing, and it will all work out. So this is lecture 3, and you'll recall last time we finished up by talking about the functions of the liver, and we were looking at table 17.7 on page 675. And as I mentioned then, I would like you to know and be able to list, be able to come up with these 7 different general functions of the liver that are shown in the left-hand column. And then also, as I mentioned, I would like you to be able to come up with, be able to write down, be able to answer a question with one specific function for each of these general functions. So this is another one of those points in the lecture at which you don't need to be writing down a whole lot of information. And let me just take a couple of minutes now and run through these general functions and the specific functions, largely just so you get some idea of all of the different types of things that the liver is doing and why the liver is so important. So general functions, and first of all then we have carbohydrate metabolism, so chemistry, chemical reactions, metabolic reactions having to do with carbohydrates. And all 3 of the specific functions you guys are familiar with already, not only from A&P1 possibly, but also from A&P2. So what's going on with carbohydrate metabolism? And now again, we're simply saying, well, again, this is happening in the liver, but glucose is being converted into glycogen when we have extra blood sugar. Just the opposite, when our blood sugar gets low, then the glycogen is broken back down into glucose, and the glucose is secreted into the bloodstream, released into the bloodstream, and gets to all parts of the body and cells throughout the body, and also the conversion of non-carbohydrates to glucose. So all of these things having to do with carbohydrates going on in the liver. Second general function, lipid metabolism. And again, I hope you're not really, trying to scramble and write down all of this stuff because I'm probably going way too fast for that to happen. So just keep in mind you want to come back to this table eventually when you are studying. So lipid metabolism again, the liver then oxidizes, breaks down essentially fatty acids. It synthesizes, it makes lipoproteins and phospholipids and cholesterol. So again, notice all of these compounds that we're talking about now are various types of lipids. It also then, the liver converts excess portions of carbohydrate molecules into fat molecules. So again, something having to do with lipids, talking about fat molecules. Protein metabolism then, a third general function. Specific functions, first of all, where it says deaminates amino acids, basically just breaking down the amino acids even further. Forming urea, which is a waste product we will talk about in a subsequent chapter. Making plasma proteins, which you may recall talking about in A&P1. And then also talking about proteins and the building blocks of proteins, which are amino acids, and so converting certain amino acids into other amino acids as the need arises for these other amino acids. A fourth general function then of the liver is storage, and specifically it stores glycogen, and we've talked about this within the last minute or two probably. It also stores various vitamins, and I don't really care that you recall which ones, that you list which ones. It stores iron, and so you may have heard people say from time to time, oh, I have low iron levels, and so I've been advised to eat liver. Um, all I can say is I sympathize. And because liver, I just can't do it. I tried liver every maybe 5 years or so just to see if maybe I like it now, and hasn't worked yet. But anyway, then, storage of iron, an important function of the liver. And finally, storage of blood, so kind of acting as a blood reservoir. A fifth basic function, then, a fifth general function of the liver is blood filtering. And recall from ANP1 then that red blood cells have a very short lifespan and a very turbulent lifespan. And basically, they're constantly being damaged and constantly being replaced. And in terms of these damaged red blood cells, then, and other foreign substances in the blood also, this filtering of these different substances, then, is a function of the liver. The next general function of the liver, then, detoxification, just removing various toxins, various harmful substances from the blood. And then finally, that last general function, secretion, and so producing and secreting bile. This then leads us into now talking about bile. And so going back to your lecture outline, and in terms then of the composition of bile, bile has different components, as the lecture outline says, and the only ones that we're really interested in here, the only ones that have a digestive function, are these compounds called bile salts. When we talk about bile, we talk not only about the liver where bile is produced, but we also talk then about the gallbladder. And notice then under gallbladder now, the gallbladder stores bile between meals, concentrates it by reabsorbing water, and then contracts to release bile into the cystic duct, as you'll recall, the duct coming away from the gallbladder. The cystic duct, again, just for review, merges with the common hepatic duct and forms the bile duct, and this then empties into the duodenum. In terms of talking then about bile release, and again, this is the release of stored bile from the gallbladder, and you can, and this figure isn't referred to on your lecture outline, but you can just take a quick look now on page 678 at figure 1730. And the gist of this is, go ahead and just start with number 1 and then follow the arrow around again. And so what's happening here in terms of regulating bile release, and specifically in terms of stimulating bile release, number 1 then in the diagram, and so chyme containing fat enters the duodenum, coming in from the stomach, of course. As we've talked about before then, this is going to stimulate the small intestine, the duodenum, to secrete CCK, this hormone that we've mentioned a couple times already. So now here's the third time that you're hearing about CCK, and it did one thing, and then it did another thing, and now here it's doing a third thing. And specifically here then, notice the CCK again gets into the bloodstream, and in this case, we're talking about, if you follow the arrow up towards the top of the page, what happens when CCK gets to the gallbladder. And as you can see then, the CCK stimulates the gallbladder to contract, and this basically forces that stored bile down to the duodenum. Talking now about functions of these bile salts, these portions of bile that, as we said, have a digestive function. So, bile salts, back to your lecture outline, bile salts aid digestive enzymes by breaking down fat clumps into smaller fat droplets This process, you'll remember maybe we went over this at the very beginning of this chapter. So this process is called emulsification. It doesn't, remember, chemically break down the fat, but it essentially breaks big clumps of fat down into smaller clumps of fat, increases the surface area, and allows faster breakdown then of the fat by digestive enzymes. Again, then specifically, the enzyme that we're talking about here that will break down the fat, that will chemically digest these individual fat molecules, these individual triglyceride molecules, that enzyme again is lipase. And again, recall then that the lipase does break the fat molecules down into their component parts. And then bile salts, back to your handout, back to your outline, also enhance the absorption of fatty acids and of cholesterol and of fat-soluble vitamins. From here, 17.8, and now we're getting into talking about the small intestine. In terms of the structure of the wall of the small intestine, first of all, if you would, off to the left where I had jotted down figure 17.36 on page 671, this should actually be page 681 if you want to flip over to that page. And then go ahead, if you would now, and flip over to page 682, and let's begin by talking about figure 1738. So again, we're talking about structures that increase the surface area of the lining of the of the small intestine specifically. You might want to jot down in your notes then that this allows faster digestion. This allows a faster rate of, and I'm sorry, I shouldn't say digestion. This increase in the lining of the surface area of the small intestine allows a faster rate of absorption, allows materials to move out of the small intestine more quickly. So, page 1730, figure 1738 then, page 682, notice that the first of these structures that increase the surface area of the lining of the small intestine are the large folds called plicae circulares. So go ahead and notice then 4 of these plicae are being shown in figure 1738. So we have these big folds. Then on the big folds we have medium folds, which are villi, medium-sized folds. Notice again they're increasing the surface area even more. And then now you can flip back over to page 681 and take a look at figure 1737, and notice here now we're looking at an individual cell along that external lining of a villus, and notice that each of these cells has even smaller folds along the medium folds, smaller folds along the villi, and notice then each of these cells has these small folds which are called microvilli. I'm going to cut off the lecture here for the moment, and we'll come back and do lecture 4 whenever you're ready.